our speakers and their excellent work and presentations. Now I'd like to briefly open this up for questions. Please state your name and affiliation before your question.
Hello, Zosha Chestakam from Medscape, and the question is for Dr. Schuster. Can I ask, what are you planning next? What sort of trials would be needed for approval of the product? And would you envisage a trial where you would compare the product to CAR T cells in this patient population? Thank you.
There's a lot of directions. I can tell you what I'm thinking in terms of immediate approach to to get this approved would be to look at the unmet need patients that have had CAR T cell therapy and are not responding, which is 2/3 in the lymphoma setting. And up until now, that's been a group of patients that we've not had much success in. So I think an easy path for development is to do a very specific study, look at the survival of patients that are failing CAR T versus those that are treated with this versus alternatives. I think it's easy. But I think, more importantly, I think that this offers a very easy way to redirect CAR T cells to bind to more than one antigen. You just heard some presentations on dual antigen targeting, and it may be a way to enhance CAR T cell effect. Well, indeed, this can be taken off the shelf and redirect CAR T cells that are genetically engineered to bind to CD19 to also bind to CD20. So maybe a very simple approach without doing any additional gene editing or gene therapy.
There's one there, and then you.
Alex Castellina from Medscape. This is a question addressed to Dr. Madhuri and Dr. Yu. Can you comment on claims of MRD made regardless of hematologic response? Typically, MRD is done in patients with stringent CR, but now, claims are made for patients with very good PR or PR. It's confusing the field, and somebody I spoke with suggested maybe new terminology is required.
I can say something first. So in terms of when we say MRD, it's not just for patients who are in stringent CR. Most patients, as you can see, went into MRD negativity, but their responses are deepening over time, and it's because it's the paraprotein. It takes a while to clear. So like an M-spike, an IgG, it doesn't clear right away, so that's why you'll see somebody may be in PR, but they're already MRD negative, but over time their paraprotein is clearing.
Question over in front.
Caroline Helwig with the ASCO Post for Dr. Madhuri and Dr. Hu. So your products both seem to be designed to persist longer and to to have a longer effect and keep patients in remission longer. How much longer do you think that will be versus, say, BB2121, for example, which is probably going to be approved soon?
So I just want to comment on the persistence. We didn't have time to touch on it, and you'll see it in the full presentation. And there's also a translational research presentation on Monday evening at 7 p.m. that talks about it in detail. Actually, only 40%, at 3 months, only 40% of the patients still had circulating CAR T cells, the CD3 positive CAR T cells. But it seemed like the persistence of those CAR T cells, or the detectability of these CD3 positive CAR T cells, did not play a role in the deepening of response.
Question over there, microphone.
Annette Brindle from BioWorld. I have a question for Dr. Valamir. In the big picture, strategically, like when would you use NK cells and when would you use T cells, and what are the advantages and disadvantages of each?
Great question. So NK cells are multifaceted. Obviously, as we know, they have activated inhibitory receptors, so they can sense a lot of different antigens at once. So we started with NK cells because of that ability, but we also appreciate the durability that T cells bring to the picture. I think ultimately we see them both being combined, and with our platform being able to reconstitute the immune system with multiple different engineered products.
Drop at BSB. Dr. Schuster, you mentioned that there were some patients who had a CR who relapsed. Do you have any theories or hypotheses as to what's happening in those patients in terms of the mechanism behind that?
Yeah. I mean, There's 4 patients on this study that had relapsed, and there was no antigen loss as is sometimes seen in CAR T cell escape. So it's not really clear. I think we need to do more work. But what I think was most remarkable is the patients responded to retreatment. It suggests that there was over time some diminution of the immune response and then the ability to re-stimulate it. It's not something that we've seen with CAR T cells.
And a quick question for Dr. Maduri. For CD19-directed CARs, we know T cell phenotypes are associated with response and this central memory. Are you creating a defined composition product? And is there some selection in the manufacturing for certain types of T cells in what you're doing?
No, so this manufacturing does not have any pre-selected. It's just we found that there's a preferential CD8 expansion over CD4.
Go ahead. Mm-hmm.
Adam Forstein with STAT. Someone alluded to BB212 in earlier question. The data from the Karma study were released last night, just a press release. But anyone up there have any comment about those data?
I mean, I can just say that every CAR T is different. What we can speak about is, for J&J 4528, it's a very different compound with 2 binding BCMA domains. It has a preferential CD8 memory phenotype than the other product. The CRS time is also 7 days, so it's completely different products, and I don't think you can compare apples to apples.
I wasn't asking for a comparison, though. I was saying we could comment on those data. If anyone wants to comment specifically on those at all.
I would say I think antigen loss is going to be a problem, and also being able to deliver the product quickly is another. So controlling tumor burden through multi-dosing strategies is probably something that you read between the line, and you see that might be an approach to overcome some of the challenges that was described.
Andrew Bowser with Internal Medicine News. I have a question about the Celian product. And PFS was reported of 78% at 9 months with a median duration not reached. And I think you said that this has a higher rate and a longer duration of response than, but I wasn't clear on what you're comparing that to. So could you talk a little bit about when you say higher, what are you comparing that to? And also, I don't recall seeing a lot of reports of effects of such agents on extramedullary lesions. So is this more, do you think this is more unique to the product, or is this something that you might see with other BCMA targeted approaches or the CD38?
In our phase 1 clinical trial, that's 8 patients out of the 9 with extramedullary tumor achieved partial or complete elimination of the extramedullary lesion. So we think that's maybe our neurotoxicity CAR T cell have effects on the tumor microenvironments. That's why we can have 5 effects on these patients.
Yeah. And the response duration, you're comparing to what? When you said it's a longer duration of response, or you're just saying in general?
Yes, compared with the previous study, most of CAR T cell target BCMA don't show this effect. That means we are the best result.
Lynn Peterson with Trends in Medicine. Dr. Schiller, could you just rub your crystal ball a little, and 5 years from now, where are we going to be in this space?
Oh, wow. Hopefully nobody's recording this, so then you can't come back and tell me what I got wrong. It's sort of like predicting who will win the next World Series. an off-the-shelf product is attractive because of feasibility issues. I actually could turn your question into a question for Dr. Schuster. How does he know that when he uses an off-the-shelf product, he is salvaging CAR T-cell response in those whose disease has relapsed after CAR T-cells? It might be some other cell population that is mediating a response. You don't know for sure. But an off-the-shelf product is always an attractive thing. It all depends on durability. An inducible pluripotent stem cell is off the shelf also. And again, that might be more desirable in terms of access, ease of preparation, and speed. So for a simple clinician, and I'm often a simple clinician, who needs to take care of patients with desperate diseases, tolerability is secondary to access and feasibility. So whatever product, be it cellular or bifunctional, that we have access to tomorrow will be better and easier for us to use. I think that's what's going to have persistence. And then you're right, the second question will be in the next phase, what's the durability? But if it only is an issue in which we can retreat and also get good response, I think clinicians will tolerate that and will accept that, and patients will accept that too. But access is very important. Okay. Well, oh, there's one more. Okay.
So, Dr. Hu, you had no neurotoxicity with your product. What do you think makes yours different from others in that regard?
Yes, it's a good question. We know that we make up this dual targeter CAR T cell targeting the BCMA and the CD38. CD38 is a long chain, but have a lower affinity with the anti-CD38. So I think this may be explain the reason why the CRS is light, I suppose.
Well, there's still one more. Okay, we'll make that the last one, I think.
Dr. Schuster, you said that you used your product in some patients who then proceeded to get CAR T-cell therapy as a bridging therapy. Is it possible that in future, while patients are desperately waiting for the CAR T-cells, to use that product as a bridging therapy in general instead of the therapies they used just now?
Yeah, well, I mean, certainly, I can tell you that having been pre-treated with this does not preclude efficacy of CAR T cells subsequently. Similarly, vice versa, I showed you data, patients that have had CAR T cells that are not working. The case in particular I was thinking of was a patient who had very rapidly paced disease, was placed on this antibody, the bispecific antibody, and had a complete remission except for one compartment. And when I biopsied that, those cells had no CD20 expression, and then went on to CD19-directed CAR and had a complete response. So this was a sequential targeting of first CD20 and then CD19 to get a complete response. I think you could do the sequence either way. This approach does not require chemotherapy. It's a chemotherapy-free approach, which is... Yeah, oh, absolutely. And particularly in responders that have not had a lot of prior chemotherapy, you can actually see increments in peripheral blood T cell counts during therapy, during the first couple months of therapy. So actually, you know, lymphopenia from prior chemotherapy is a major obstacle to CAR T cell production, but getting cells, enough of them in cells that are functional. So this could be a bridging approach that doesn't require chemotherapy, that actually stimulates and invigorates T cells, and then can be used to make a CAR T cell product.
All right, well, thank you to the investigators who presented us their potentially practice-changing results, and thank you all for your attention.